Okay, let's move on. We're going to look at continue on with chapter 18. Looking at the blood, and this is video 2 of 3. And we're going to deal with the erythrocytes. Now here are the objectives you should know at the end of this section, such as the structure and the function of the erythrocytes and the hemoglobin, the life cycle of the erythrocytes, naming and describing the types, causes, and effects of the RBCs. Those are deficient or in excess of these RBCs. So erythrocytes, also known as the red blood cells, have 2 types of principles: to pick up oxygen from the lungs and deliver it to tissues, and to pick up carbon dioxide from the tissues and unload it into the lungs. Now, a lack of erythrocytes, as may occur with major trauma or hemorrhage, can be fatal within a few minutes due to oxygen deprivation. Now, in terms of form and function, an erythrocyte is a discoidal cell with a thick rim and a thin sunken center, about 7.5 micrometers in diameter and 2.0 micrometers thick at the rim. The red blood cells lose nearly all organelles during their development and are less remarkably devoid of internal structure. Having no mitochondria, they rely exclusively upon anaerobic fermentation to produce ATP and are the only cells to be so indefinitely. They do not consume oxygen that they carry. Now, the cytoplasm of an RBC consists mainly of 33% solution of hemoglobin, about 280 million molecules per cell. This red pigment gives an RBC its color and name. Now, hemoglobin transports oxygen and also aids in the transport of carbon dioxide and the buffering of pH. The cytoplasm also contains carbonic anhydrase, an enzyme that catalyzes the reaction, changing carbon dioxide to bicarbonate in the presence of water. The plasma membrane of a mature RBC has glycolipids on the outer surface that determine the blood type. The inner surface has 2 skeleton proteins, the spectrum and the actin, that give the membrane resilience and durability. Resilience is important when RBCs pass through small blood capillaries and the sinusoids. These passages are smaller than the diameter of the RBCs, and they must stretch, bend, and fold so they squeeze through. Hemoglobin consists of 4 protein chains called globins. Now, 2 of these, let's pull up the whole thing for you guys. Now, 2 of them are alpha chains, and they're approximately 141 amino acids long. And the remaining 2 are the beta chains, and they have approximately 146 amino acids. They are 146 amino acids long. Now, each chain is conjugated with a non-protein moiety called the heme group, which binds oxygen to F2, which is our ferrous ion, at its center. Let me go back, sorry. And each heme carries 1 oxygen. Now, about 5% of the carbon dioxide in the blood is also transported by hemoglobin, but is bound to the globin moiety rather than the heme. Now, the hemoglobin exists in several forms with slight differences in the globin change. We have an adult, which HbA, and we have fetal, HbF. Now, the adult hemoglobin is the most common, but about 2.5% of an adult's hemoglobin is HbA subscript 2, which has 2 delta chains in place of the beta change. With the fetus produces a fetal hemoglobin, which is the HbF, which has 2 gamma chains in the place of the beta change. So our HbF binds oxygen more tightly than our adult hemoglobin. The RBC count and hemoglobin concentration determine the amount of oxygen the blood can carry. The hematocrit, packed cell volume, is the percentage of whole blood volume composed of red blood cells. The range in men is normally 42 to 52%, and in women is 37 to 48%. The hemoglobin concentration of whole blood is normally 13 to 18 grams per deciliter in men and 12 to 16 grams per deciliter in women. The RBC count is normally 4.6 to 6.2 million RBCs per microliter in men and 4.2 to 5.6 million per microliter in microliters in women. The differences in values between men and women are due to 3 physiological reasons. Androgens, they stimulate the red blood cell production. The women of reproductive age have periodic menstrual loss of blood, and the hematocrit is inversely proportional to percentage of body fat, which is higher in women. In men, blood also clots faster, and the skin has fewer blood vessels, traits that may have an adaptive value. And erythrocyte lives for an average of 120 days from production in the bone marrow. Now, erythrocyte production is called erythropoiesis. Let's see if I can spell that here for you guys. I don't remember if I have it on one of the other slides, but it's spelled E-R-Y, sorry for my writing, T-H-R-O-P-I-E-S-I-S. And it normally takes approximately 3 to 5 days for this process or for this production to occur. Now, it begins when the hematopoietic stem cells in the bone marrow becomes an ECFU, which is erythrocyte colony forming unit, which has receptors for the hormone erythropoietin. Now, the ECFU, upon stimulation by EPO, transforms into erythroblasts, which are those normoblasts. Now, once those EPOs transform into the erythroblasts, it multiplies and it synthesizes the hemoglobin. Now, when this task is completed, the nucleus shrivels and is discarded from the cell, which is now called reticulocytes. It contains a temporary network of ribosome clusters. Now the reticulocytes, they leave the bone marrow, and in a day or two, the last of the ribosome clusters to disintegrate, and the cell is a mature erythrocyte. So here you see in this particular diagram everything that I was just referring to, where you have the erythrocyte colony forming unit, which has a receptor for that protein. Now when it's stimulated by the EPO, which transforms into the erythroblast. And this is when it starts to multiply and help to synthesize that hemoglobin. And after that is completed, the nucleus starts to shrivel, and then it's discarded out of the cell, and then it's now a reticulocyte, where you see all these clusters of ribosomes inside of this reticulocyte. Now when this leaves this bone marrow here, after what, a day or two, the last of the ribosomes, the clusters, they disintegrate. And this is where we have our mature erythrocyte. So let's talk about iron for a minute. Iron, most of iron within the body is found in hemoglobin, which we already should know, within erythrocytes. Iron is stored in the macrophages, and in a lesser extent in the hepatocytes, which represents the storage pool of iron. Now, small amounts of iron are found in myoglobin and in plasma, and those are the ones that are bound to the transferrin. Now, a typical adult human body contains about 3,000 to 4,000 milligrams of iron. Only about 1 milligram of iron is lost from the body per day, and that's through blood loss or a sludge mucosal epithelial cells, and most can be replaced through diet. The majority of iron required by the body is acquired by recycling iron from senescent, and those are like aging cells, aging red cells, where they're either aging or they're no longer capable of dividing, but they're still alive and metabolically active. So we know that it's a very crucial nutrient requirement for our body. Now we consume approximately 5 to 20 milligrams per day. Now women require more iron than men due to menstruation, and a pregnant woman needs almost twice as much in the last 3 months of pregnancy. Now, dietary iron may be ferric, which is our F3, or ferrous, which is our iron 2. Ferric is Fe3, and ferrous is Fe2. Those are particular ions. Now, the stomach acid converts ferric to ferrous, which is the only form that can be absorbed. So let's take a look at this diagram. We'll talk a little bit more about how iron is metabolized. Now, once we ingest iron, it needs to be broken down. It mixes and we ingest it, and it reaches through the digestive system. It reaches to the stomach. Now when it reaches the stomach, we have a protein called gastroferritin. You see right here, the gastroferritin, which is what the stomach produces, and it will bind to the Fe2. Because remember, F3 is converted into F2 when it hits the stomach. And the stomach acids is what's converting it. So now we have the F2 binding with the gastroferritin. And this is what helps transport it to the intestines. Now, once absorbed in the blood, the F2 binds to a plasma protein called transferrin, which carries it to the bone marrow, the liver, and other tissues. So here we have our transferrin, which is our purple triangles binding with our with their Fe, and this is going to help take it to where? The bone marrow, the liver, and other tissues. Now, bone marrow uses the Fe2 for hemoglobin synthesis. Muscles use it for myoglobin, and nearly all cells use it in cytochrome molecules in the mitochondria. Now, the liver binds surplus iron to apoferritin to produce an iron storage complex called the ferritin. Now, other nutritional requirements for erythropoiesis includes vitamin B12, folic acid, vitamin C, and copper. Now, the vitamin B12 and the folic acid, their responsibility is to assist with rapid cell division and DNA synthesis that will occur in the erythropoiesis. And if we have a deficiency, it will lead to a production of nucleated red cell processors. Now, intrinsic fibers from the stomach is required for absorption of vitamin B12 from the terminal ileum. Now, vitamin C and copper, these are cofactors for enzymes synthesizing hemoglobin. Vitamin C is like a reducing agent. It facilitates the absorption of iron from the gut. It is involved in the normal metabolism of vitamin B12 and folic acid. Erythrocyte homeostasis, namely the red blood count, is maintained by a negative feedback loop. Now, if the RBC count drops if the RBC count drops due to someone hemorrhaging, it can result in hypoxia. The kidneys are going to detect the hypoxia, or detect that, detect the hypoxia, and then increase the EPO to be released. Then the red blood cell count rises 3 or 4 days later. Now, hypoxemia may result from causes other than blood loss. But travel to high altitudes may produce temporary hypoxia. People who live at high altitudes have higher RBC counts. Now, an abrupt increase in oxygen consumption might also cause hypoxemia, which we have like endurance trained athletes have a higher red blood count, a red blood cell count. Now, in each case, hold on one second. Okay, so in cases such as emphysema, hypoxemia cannot be corrected by increasing the red blood cell count, but the body attempts to increase it anyway, and this leads to a dangerous excess called polycythemia. Alright, so let's move on to erythrocyte death and disposal. Now, after a while, the membranes of the erythrocytes start to get fragile, and then they eventually die and need to get disposed of. Now, many RBCs die in the spleen, where old RBCs become trapped. Hemolysis, the rupture of red blood cells, releases the hemoglobin and leaves empty plasma membranes, which are discarded by the macrophages. The hemoglobin disposal is critical since it can block kidney tubules and cause kidney failure. Now, the macrophages, what they'll do, they start to separate the heme from the hemoglobin. So you have this hemoglobin, and then they'll take away the heme they'll separate the heme from the hemoglobin. Then they're going to hydrolyze the hemoglobin into amino acids. The macrophages then remove the iron from the heme and then release it into the blood, where it combines with the transferrin. Now, the rest of the heme is converted by the macrophages into a greenish pigment called biliverdin, biliverdin, and then to a yellow greenish pigment called bilirubin. Now, bilirubin is released, and then it binds to the albumin in the plasma. The liver then removes it and secretes it into the bile, which is discarded into the small intestines via the gallbladder. The biliverdin and the bilirubin are called bile pigments. The bacteria in the small intestines, they play a good role. They help to break down the bilirubin into urobilinogen, which has a brown color. Another pigment, urochrome, produces the yellow color of urine. Now, high level of bilirubin in the blood causes jaundice, which must be a sign of liver disease, rapid hemolysis, or bile duction obstruction. Now, an imbalance between the rate of erythropoiesis and the RBC destruction may lead to erythrocyte disorders. We have polycythemia, which is a disease of excess red blood cells. We have primary and secondary polycythemia. The primary one is due to cancer of the erythropoietin line of the red bone marrow and can result in an RBC count as high as 11 million cells per microliter. Now, remember, I said that the normal amount for men is 4.6 to 6.2 billion, and for women is 4.2 to 5.4 million. Now, that's a huge increase. And of course, the hematocrit as high as 80%. Now, as with the secondary polycythemia, it's a term used in cases. Let me pull these up for you guys. Is used in cases of polycythemia from all other causes, such as dehydration and high altitude. And as red blood cell counts as high as 6 to 8 million cells per microliter. So that's almost double the amount. Now, the primary dangers that we'll have with polycythemia are increased blood volume, pressure and viscosity, and then with chronic polycythemia, it can lead to embolism, stroke, or heart failure. Now let's look at anemia. Anemia is a condition that develops when a blood lacks enough healthy red blood cells or hemoglobin. And here we have a lot of the symptoms that can occur. I found this on the internet, and I thought it was neat just to put it in there so you see the various symptoms. Because some people just think, oh, this person's just cold. That's just one sign. But in extreme, that's not the only one. But in extreme cases, you'll have a person suffering from chest pain or angina, or even from a heart attack, or even fainting. So some other ones that are not severe, we have fatigue, we have dizziness, low blood pressure, palpitations, lethargia, as well as weakness in the muscles, discolored stool supposed to be a certain color, and if it's not that color, then it's a possibility it can be person can suffer from anemia. Then we have yellowing of the skin and as well as the eyes. And of course we have at the bottom here showing you enlargement of the spleen. So let's take a look at this this chart and exactly what causes it. Anemia has causes that fall into 3 categories. We have inadequate erythropoiesis. There is blood loss, which is our hemorrhagic anemia, and then we have our hemopoietic anemia from red blood cell destruction. Now, under each, well, the first and the last category, you notice they have various examples of what they're referring to. We have anemia of old age. We have the declining of the erythropoiesis due to nutritional deficiencies, a reduced amount of physical activity, gastric atrophy, and renal atrophy. There's also anemia due to renal insufficiency, where it's caused by, so for example, deficiency of the EPO secretion, or even deficiency or lack of thereof of various nutrients, such as folic acid, vitamin B12, and vitamin C, and of course iron, which is our iron deficiency anemia. Then on the last one, we have red blood cell destruction, where we have drug reactions, such as penicillin allergy. We have parasitic infection with our RBC destruction by the malaria parasites. Sorry. Alright, so I already gave you some of the examples of I showed you guys the chart of the 3 categories that it falls into. And here's just a little more information, written information. Don't forget to look at your textbook and write down or include additional information. Please do so. Then we have anemia. Has 3 potential consequences. It has tissues often suffer from hypoxia. That can lead to necrosis of the brain, the heart, and as well as the tissues. The patient can suffer from lethargia, shortness of breath with exertion. So as any person that may walk, simple things such as walking up the stairs, maybe just walk to the car, walking to the kitchen. The person is short of breath, trying to gasp for air. Then we have blood osmolarity is reduced, leading to edema. and blood viscosity is reduced, causing increased heart rate and low blood pressure. Eventually, cardiac failure can occur. Sickle cell disease, another disorder. Sickle cell disease is a hereditary hemoglobin defect that occurs mainly in people of African American descent. Sickle cell disease afflicts about 1.3% of people of the African American descent and is caused by a recessive allele that substitutes valine for the glutamate acid and the 6th amino acid of the hemoglobin beta chain. Now, people who are homozygous for the hemoglobin HbS, it exhibits that the person may have the disease. No, actually will have disease. People who are heterozygous for this particular sickle cell trait, they very rarely have severe symptoms, but they do have the trait. So if they would mate with an individual who is homozygous, that means they do have the disease. It is a strong possibility that their offspring will as well have the disease, or half will have the disease and the other half may not. So hopefully you guys kind of remember that from maybe high school, about your Punnett squares, understanding the various traits that they passed on. So then if we have two heterozygous carriers that reproduce, their child has a 25% chance of being homozygous. Now, if the hemoglobin sickle cell trait or the HbS does not bind oxygen, well, it does not bind oxygen well, and at a low oxygen concentration, the erythrocytes become elongated and then pointed at the ends, which you see right here. So they become elongated, and here we have our pointed ends. Now, the sickled erythrocytes are sticky and clumped, and they block small blood vessels, causing intense pain. Now, the blockage of the circulation can lead to kidney and heart failure, stroke, rheumatism, as well as paralysis. Now, hemolysis may cause anemia and hypoxemia resulting from further sickling. Now, chronic hypoxemia also causes fatigue, weakness, mental deficiency, and organ deterioration. Hemopoietic cells become enlarged and misshaped. Sickle cell disease is an example of pleiotropy, the existence of multiple effects of a phenotype from a single gene change. And without treatment, a child with sickle cell disease has little chance of living to age 2. Now, with treatment, one can live a decent life a little beyond the age of 50. It is thought that the heterozygotes have resistance to malaria because their blood cells will not support the parasite that causes the disease, and it is the result, and it is the reason for gene persistence. So here we have this gentleman here, James Drew. He's the first black person to pursue advanced degree in medicine to study transfusion and blood banking. Now, he used plasma rather than whole blood, and it caused less transfusion reactions. Please remember Charles Drew. You will definitely see him again on a test. Our other person here is Carl Langston. He discovered that blood types A, B, and O back in 1900, and he won the Nobel Peace Prize in 1930. Now the type AB was discovered a lot later. Now, blood types are based on interactions between antigens and antibodies. Okay, I sort of mixed up my slides, so I have to rearrange them. So, with blood types, okay, blood types, they're based on the interactions between antigens and, let's get to that slide, between antigens and antibodies. Antigens are complex molecules such as proteins, glycoproteins, and glycolipids that are unique to each individual. When the body detects an antigen or foreign origin, it produces an immune response. Part of this response is the release of proteins called antibodies. Antibodies, they bind to the antigens and they destroy them. One method is agglutination, in which each antibody molecule binds two or more antigens, producing antigen-antibody complexes. Now, blood types are based on antigens called agglutinogens on the surfaces of the red blood cells, and the antibodies called agglutamins. And those are the ones that you'll find in the blood plasma. These react to the glutinate red blood cells in the event of a mismatched transfusion. So our RBC antigens, they're called agglutinogens. Sorry. And they're called antigen A and antigen B, and they're determined by the carbohydrate moieties found on the red blood cell surface, like I mentioned earlier. And then we have the antibodies. Those are the ones that are called agglutinins, and they're found in the plasma, and they're anti-A's and anti-B's. It's just a diagram. I'm not going to go into too much details with this one, so don't worry about this slide. Now, agglutination is each antibody can attack to several foreign antigens on several different red blood cells at the same time. Now, responsibility for mismatched transfusion reaction. We have agglutinated red blood cells block small blood vessels, like I mentioned already. And what is it called? It causes other complications. And if, of course, they're not taken care of, probably it will cause kidney tubes to be blocked, and of course, renal failure. Right, so our AB group consists of group types A, B, AB, and O. Now, a person's ABO blood type is determined by the hereditary presence or absence of the antigen A and B on the person's red blood cells. Antigens of ABO group begin to appear 2 to 8 months after birth, and they reach maximum concentrations between 8 to 10 years of age, and then they slowly decline. Our antibodies of the AB group react against the A or B antigen, except those on one's own RBCs. The antigen that reacts against the A is called the alpha, just say anti-A. The anti-A, and is present in plasma of people with type O or type B blood. The antibody that's against antigen B, those are considered to be anti-B, and is present in the plasma of people with type O and type A. Each antibody molecule has 10 binding sites and can therefore attach to several red blood cells at once and agglutinate with them. The most common type is type O, and our rarest is a type AB, sorry. So in this diagram, in this figure 18.15, this is entitled The Effects of Mismatched Transfusion. This is when a donor's RBCs become agglutenated in the receptor's blood plasma. The agglutenated RBCs, they start to enlarge in smaller blood vessels, which you see right here. So to enlarge in the smaller blood vessels downstream from this point, and then they'll cut off blood supply to any of the tissues. So if a transfusion reaction occurs, for example, if type B blood is transfused into type A recipient, the recipient's anti-B antibodies would cause immediate coagulation of the donor's red blood cells. Now type AB is sometimes called the universal recipient because the blood lacks both anti-A and anti-B. However, the donor's plasma may agglutinate the recipient's red blood cells if it contains anti-A and anti-B, or even both. Now type O is sometimes called the universal donor, and that's because the red blood cells do not contain A or B antigens. However, the plasma of type O donor can agglutinate the red blood cells of A, B, or AB recipients. Let's pause. Right, one thing is that blood type is not changed by transfusion. The type is mixed at conception. Now when we look at the Rh blood group, it includes numerous RBC antigens, of which the principal types are C, D, and E. Now antigen D is by far the most reactive, so a person is considered an Rh positive if he or she has the D antigen and Rh negative if lacking the antigen. Now ABO blood type has no influence on Rh type or vice versa. The Rh frequencies vary among ethnic groups, just as the ABO frequencies do, but they vary independently. Anti-B antibodies are not normally present in the blood. They form only in Rh negative individuals exposed to Rh positive blood. Alright, so we're going to look at a diagram on the next one of the other slides that I have here. Is going to show you an example of this Rh. So if we have, I'll show it to you, let me see, right here, I believe it's on figure 1816. So if you see it in your book, you can follow along, because I'm going to go back one slide. Now if an RH person receives an RH transfusion, he or she can produce anti-D, which will present danger if another RH positive transfusion is received. Now a related condition occurs when an RH negative woman bears an RH positive fetus. Let's go to that page here. So if the mother is RH negative and receives an RH positive transfusion, the fetus will receive that RH positive, will be RH positive. Now the second pregnancy with an RH positive fetus may result in the fetus being affected by the anti-D in the mother's placenta, which passes through the let me try that again. If the second pregnancy with the RH positive fetus, it may result in a fetus being affected by the anti-D in the mother's plasma, which passes through the placenta. Now the newborn, the baby is born with a severe anemia called hemolytic disease of the newborn, HDN. Now not all HDN is due to this RH incompatibility. Only 2% of the cases result from an ABO or other blood type incompatibility. Now the HDN, which we already said is the hemolytic disease of a newborn, is easier to prevent than to treat. If an RH mother can be given an RH immune globulin during the pregnancies, it will bind fetal RBC antigens, so anti-D is not produced. So we don't want this anti-D. The infants with HDN are often severely anemic. Their tissues produce and release immature red blood cells in an effort to offset the anemia. Now high bilirubin levels from hemolyzed RBCs can produce a particular syndrome that's very toxic to the brain, which is called, let's see if I can remember the name, Kernicterus, Kernicterus. See, I'm not going to try and spell it for you, write it down for you guys. My spelling was not nice. And of course, with the HDN, it can be treated with a phototherapy where it is exposed to UV rays, which degrade any type of the bilirubin as it passes through the skin capillaries. Now, an exchange transfusion may replace the infant's RH positive blood with R negative, allowing time for the mother's antibody to disappear from the infant's blood. So that's the end of our chapter 13, part 2 of 3. So we have one more to look at, and we're going to look at the white blood cells.